Dear students, in this session we will learn about the structure of Polytene chromosomes. Now these chromosomes are special chromosomes wherein they exhibit huge size when compared to all other normal type of chromosomes, and because of its larger size it is called as Gaint chromosomes. Now this picture shows you this is how the Polytene chromosomes look like when it is stained, and this is the salivary gland chromosome of Drosophila. Now these chromosomes were first seen in the salivary gland cells of dipteran species like in Drosophila and in Chironomous, and because of which it is called as salivary gland chromosomes or Polytene chromosomes. Now the main objective of this session is to make you understand the structure of Polytene chromosomes, and by the end of the session I am sure you will all appreciate the complexity of this structure of Polytene chromosomes and its significance. Now we will discuss this topic under these headings. We will learn about the occurrence of these Polytene chromosomes, their nature, how they are formed, and their structure, which includes the bands and the interbands, and the chromosomal puffs and the Balbiani rings, and finally the functions of these Polytene chromosomes. Now, where do you find these Polytene chromosomes? They are basically found in the cells of salivary glands of Drosophila and Chironomous larva, and in fat bodies of certain dipteran larvae. It was first discovered by E.G. Balbiani in the year 1881. Now these Polytene chromosomes, they are multi-stranded, hence the name Polytene. Poly means many, teen is the fibers or the strands. So since the structure of this Polytene chromosomes is made up of many number of strands, it is named as Polytene chromosomes. And these chromosomes may reach a size that is up to 200 times or more the size of the corresponding chromosomes at meiosis or in the nuclei of the ordinary mitotic cells. So their size will be 200 times larger than the ordinary chromosomes. Now, what makes these chromosomes multi-stranded? How this multi-stranded nature is formed? Here, these Polytene chromosomes are formed with a specialized process called as endoreduplication. Now these Gaint chromosomes, they present a bundle of fibrils which arise by repeated cycles of endoreduplication of single chromatids. Now here endoreduplication means that the chromatin replicates without cell division, as a result of which the number of chromonemeta keeps on increasing. Hence, these chromosomes are popularly known as Polytene chromosomes, and the condition is described as polyteny. And the number of chromonemata fibrils per chromosome may reach up to 2000 in extreme cases, and some workers have also reported as high as 16,000 fibrils which is observed in certain Polytene chromosomes. Now let us understand the structure of these Polytene chromosomes. These Polytene chromosomes, they present a distinct pattern of transverse banding which consists of alternate chromatic and achromatic regions, that is stained and the non-stained, or the darkly stained and the lightly stained regions. These bands, they greatly helped in the mapping of the chromosome in the cytogenetic studies. So the ones which are darkly colored are called as dark bands, which are rich in DNA and it has less amount of RNA in it, whereas the other bands which are lightly stained, they are called as interbands, and these interbands, they show high concentration of RNA and lesser amount of DNA in it. Now these bands are specific for a particular chromosome, and banding pattern is almost similar in both the homologous chromosomes. Initially it was believed that each band represents a gene, but now it is estimated that the average band contains about 30,000 base pairs, or 20 times the amount of DNA required to code for an average protein, and hence it is polygenic. Now this picture explains you the structure of a polytene chromosome in Drosophila melanogaster. So this is stained or mapped from the salivary gland of Drosophila melanogaster. Here this polytene chromosome, it is found in the form of 4 long and 1 short limb radiating from a single, more or less amorphous mass known as chromocenter. So it is in this region of chromocenter, or centromeres of all these 4 chromosomes are attached here. So all the limbs originate from this chromocenter. One long limb corresponds to the X chromosome, and the remaining 4 long limbs correspond to they are the arms of the second and the third chromosome. So in the figure you can notice there are 2 arms, the right arm and the left arm. So the right arm of the second chromosome is normally referred as 2R, and the left arm of the second chromosome is referred as 2L. Similarly, 3L means left arm of the third chromosome, and 3R means the right arm of the third chromosome. The centromeres of all these chromosomes fuse to form the chromocenter. So in the male flies, the Y chromosome is also found fused within the chromocenter and is therefore not seen as a separate strand. Bridges proposed one reference system in which the limbs of the salivary glands are divided into 102 sections, which are called as divisions, and each division is designated by a number 1 to 102. So each of the 5 main limbs, so which are the 5 limbs here, the X chromosome, 2L, 2R, 3L and 3R, contains 20 divisions. The short chromosome 4 contains only 2 divisions. Each division is represented by a prominent band and is divided further into 6 subdivisions A to F. For example, we write 16A for the barred eye locus. So which means the gene is located at the 16th division and A stands for the subdivision. Thus, each individual band of salivary gland chromosomes can be identified by giving the division number, subdivision designation, that is from A to F, and the number of the bands starting from the beginning of the subdivision. Bridges presents the following minimum numbers of bands for the salivary gland chromosomes of Drosophila melanogaster. So here suggested 537 bands for the X chromosome, 1032 bands for the second chromosome and 1047 bands for the third chromosome and 34 bands for the fourth chromosome, totaling a minimum of 2650 bands for the whole genome of this Drosophila melanogaster. Now, when you observe this structure of Polytene chromosomes, here and there, apart from the light band and the dark band, you can see certain regions of this chromosome shows swelling or little bulged up. So, this bulged portion of the chromosome arm is what is called as chromosomal puffs. Chromosomal puffs. Now, certain region of this gained Polytene chromosome is modified due to enlargement and extension of bands into lateral loops called as Balbiani rings or the chromosomal puffs. So they are presumed to be formed by unwinding and unrolling of chromonemeta, which otherwise is closely folded or coiled in the bands. Now these chromonemata project into series of loops which increase the thickness or the diameter of the chromosome and hence provide fuzzy appearance. The formation of puffs is controlled by specific genes and takes place at definite time. Puffs contain a high amount of RNA and is chiefly associated with the metabolic activities of the chromosomes because of high RNA concentration. Now this diagram shows you the oversized puff which results in the formation of Balbiani rings. And you can see the looping out of chromonemata as well. Now this is the whole diagram that summarizes the structure of a Polytene chromosomes. Here you can see the dark bands, the light bands which are alternating with each other, and here and there you see the puffs being formed, and in certain regions the puffs become too enlarged, giving rise to the Balbiani rings. And when you zoom in these Balbiani rings and see, it shows looping of these chromonemata, which in turn further contains loops with all mRNA synthetase being present on each of the spiral. Now these puffs, let us learn like what these puffs are made up of and how or what is the importance of these puffs. Now puffs consist of DNA loops that are less condensed than the DNA of bands elsewhere in the chromosome. The puffs are active sites of gene transcription. The condensation and the decondensation at puffs correlate with enhancement of transcriptional activity at these sites. The process of puffing involves several processes such as accumulation of acidic proteins, despiralization of DNA, accumulation of RNA polymerase II. We all know that it's an enzyme involved in the transcription of messenger RNA molecules and also in the synthesis of mRNA, and the release of newly synthesized mRNA in the cytoplasm. Now, what are the functions of these Polytene chromosomes? The main function of the Polytene chromosomes is to carry genes which ultimately control the physiology of an organism, and these genes are formed of DNA molecules. Secondly, it assists in the shifting of heterochromatin in respect to euchromatin, which produces gain changes leading to position effects, and because of these position effects it results in mutations, which in turn brings about variations. Heterochromatic regions contain fewer genes than the euchromatic parts, and the production of nuclear material is entirely done by heterochromatin. And lastly, The chromosomes, they help in protein synthesis indirectly, and the nucleolus contains RNA, and this RNA serves as a means of transmission of genetic information to the cytoplasm, leading to the formation of specific protein. So this is how these Polytene chromosomes functions. You can refer to these following textbooks for further more details. You can refer to Genetics by Virbal Rastogi, Textbook of Genetics by Verma and Agarwal, or you can also refer to the genetics textbook written by P.K. Gupta. Now here are a few questions to ask. Just to take a recap of what you have learned or how much you have learned. Chromosomes found in the salivary gland of Drosophila is called as option A, Polytene, B, lamp brush, C, supernumerary, and D, B chromosomes. So what's your answer? Your answer should be Polytene chromosomes, so it is option A. The second question, the Gaint chromosome with a number of chromonemata is called what? Heterochromosome, Polytene chromosome, lamp brush chromosome, or supernumerary chromosome. So what's the answer for this? It is answer B, Polytene chromosome, because we said that the chromonemata keeps on dividing in spite of the once the cell division stops. So because of that endo reduplication, you have many fibers of this chromonemata being formed, and hence it results in Polytene chromosomes. So puffs are the Balbiani rings in the salivary gland. Chromosomes are the sites of protein synthesis, RNA synthesis, DNA replication, or DNA duplication. So what's the correct answer? The answer is they are the sites of RNA synthesis. So the right answer is option B. Then the fourth one, the point at which Polytene chromosomes appear to be attached together is known as a centriole, a chromocenter, or centromere, or the chromomere. What's the right answer? It is a